This call is now being recorded.
Okay, can you see the screen? Can you see the screen? Just unmute yourself and let me know.
Yes, ma'am.
Yeah, okay. So, last time in the last lecture, what we discussed was all these terminologies and double fertilization. Okay, we discussed up to double fertilization, as well as we discussed the contrivances for the self cross pollination, difference between the self and cross pollination. So all these things we have already discussed. Now we'll come to the next part, that is the significance of double fertilization. Now what does it mean, that double fertilization? Because twice this fertilization takes place, both the male gametes are utilized, so that is called as double fertilization, which is the unique feature of angiosperms. It ensures that the parent plant invests a seed with a food store only if the egg is fertilized. So this is the most important thing. Under significance, you should write that's a unique feature of angiosperms, and seeds which are produced with the which store food. So this is one of the importance and significance of double fertilization. Then the second is diploid zygote develops into an embryo, which consequently develops into a new plant. As you know that the result of first fertilization is what? Formation of zygote. And this is the first diploid cell. Then zygote in the further process of development gives rise to embryo, which gives rise to a new plant. So this is very important. Then the triploid primary endosperm nucleus develops into nutritive endosperm tissue. That means what? The second male gamete, when fuses with the secondary nucleus, then what happens? Or that triple fusion, or you can see whatever the result of this second fertilization is triploid primary endosperm nucleus, which acts as a nutritive tissue for the development of embryo, which is producing, which is developing, which is the result of first fertilization. Zygote gets develops into embryo. So for that embryo, this endosperm, triploid primary endosperm, works as the nutritive tissue. Okay, so this is also very important. Then it restores the diploid condition by fusion of haploid male gamete with haploid female gamete through syngamy. That means you know that syngamy is what? First fertilization. One of the male gametes fuses with the female gamete. That means egg cell. Both are haploid. So what happens then? The result of the fusion of two haploid gametes is diploid condition. That is zygote is formed. So this condition is restored again, which is very important. Then it also helps to avoid polyembryony. Now what exactly polyembryony we will discuss in the later part. So it avoids polyembryony. Poly means many embryos are developed. In some cases, in some species, many embryos are developed which are not helping the uh, development of that particular seed, or you can say the at a time many embryos are produced that can be avoided with the help of this way of double fertilization. This is what you remember just as significance. We will discuss this part polyembryony in the later part of this chapter. Now as we were talking the significances of the double fertilization, now this is the first thing. Now we have come to, that is endosperm. What is that endosperm? As we discussed right now, the second male gamete when fuses with the secondary nucleus, then the what result of this fusion is what? Triploid primary endosperm nucleus. So this is what called as TPEN, and this is very important as the nutritive tissue in the developmental stages of embryo. So mitotically to form nutritive tissue called endosperm, that is the result of the second fertilization. There are different types, three types we are going to discuss, which are involved in our syllabus. That is nuclear endosperm, cellular endosperm, and halobial endosperm. What are these three types of endosperms? How are these produced or how they look? First, nuclear endosperm. This is the most common type found in 161 angiospermic families. And the primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nuclei. A big central vacuole appears in the center of cell, pushing the nuclei towards the periphery. Now, what you understand by this? Just try to understand that nuclear means what? Whatever the endosperm is produced, that divides mitotically. That means TPEN. That is triploid primary endosperm nucleus, which is the result of second fertilization. That divides mitotically without formation of wall around the nucleus. That means what? Many nuclei can be seen without any wall, so that it gives the appearance as many nuclei in a single cell, right? So this is what called as nuclear type of endosperm, where a big central vacuole appears in the center of cell, pushing the nuclei towards the periphery. So that means at the center, that cell is there and the nuclei are shifted towards periphery. I will show you this with the help of diagram later. Just try to understand. A second type is what? And later in the later stage, that each nuclei is followed by the formation of wall also, but in the later stage. And the example of this type of nuclear endosperm is wheat, sunflower, coconut, etc. Cellular means exactly what happens the same. Means the primary diploid primary endosperm nucleus divides mitotically. After the nuclear division, it is immediately followed by the cytoplasmic division. That means each nucleus is surrounded by the wall. So this is called as, which gives the appearance of many cells. So that is what called as cellular type of endosperm. And right from the beginning, that nucleus is followed by the formation of wall around itself. That means cells, many cell structure it gives the appearance as. So balsam, petunia, adoxa, these are the examples of cellular type of endosperm. Next is allobial type of endosperm. The name has come from the family of the name, the order you can say of the monocotyledons, that is Halobiales, and Halobiaceae is the family, where because of which the name is there, that is Halobial type of endosperm. That is considered as a combination of first and second. That means nuclear and the cellular type of endosperm. That means what happens here? First of all, it is followed by the transverse wall. That means first division is transverse wall formation, which divides the cell unequally. That means from that one single cell structure, transverse wall is formed between these, so that endosperm, triploid primary endosperm, divides unequally. And smaller cell is called chalizial cell, and larger cell is the micropilar cell. That means the unequal cells, smaller one is towards the chalizial end and larger one is towards the micropilar end, right? So first is the transverse division, which is unequal. Smaller is towards chalizial end, larger is towards the micropilar end. Then the nuclei in each cell divide by free nuclear divisions, and then walls develop between nuclei in micropilar chamber. That means what? The smaller one, which is towards the chalizial end, that divides again mitotically with free nuclear divisions, like first type, nuclear type of endosperm. And larger cell, which is towards micropilar end, that also divides mitotically with free nuclear divisions, but the wall formation is there in the larger cell immediately after the nuclei are formed. So that means what? It is considered to be the intermediate stage or the type of the cellular and nuclear type of endosperm, because both the characters are seen in this type of allobial type of endosperm. Asphodelax is the example of this type. Many members of the family, this family, hello BHC, all they show such type of endosperm. We have discussed about the endosperm. Now we will talk about the embryo. Try to imagine, you keep this diagram in front of you of double fertilization, so it will be easier for you to understand. The diagram of double fertilization 1.6 anatropous ovule is there. Then if you keep the diagram of double fertilization in front of you, that is the 1.13, then you just try to understand the result of first fertilization and second fertilization. First fertilization is what? Formation of zygote. And second fertilization is what? With the secondary nucleus, that is TPEN, right? Now we spoke just now about the TPEN development. What happens to TPE? Endosperm is produced. And in these three categories, types of endosperms are seen. Now we are talking about the first fertilization result, that is zygote, right? Now what happens to that zygote? The process of development of zygote into an embryo is called embryogenesis. Always remember, if you split this word, genesis is what? Generation. What is generated? Embryo is generated. From what it is generated? From the zygote it is generated. What is the zygote? That is the diploid cell. How the zygote is produced? By the fusion of male and female gamete, that is called as syngamy or first fertilization. So this is what you need to remember. And where it is taking place? That is taking place towards the micropylar end, isn't it? Because the first fertilization is taking place near the micropylar end, where female gamete is near the micropylar end, and that male gamete reaches over there to form the zygote. So imagine that diagram, that where everything is taking place, right? So this development of embryo, now we will talk about in detail. So zygote, which gives rise to embryo, that is taking place near the micropylar end, whereas TPEN, second fertilization result, that is TPEN, give rise to endosperm, that is towards the chaliceal end. Now what happens to this first, that is zygote? Zygote, it forms a wall around itself and it is called as oospore. It looks like a spore, and oo word is generally related to the egg-like, because egg only by the fusion of male gamete got converted into zygote, right? So this is what you just remember, spore-like structure. So zygote, it forms a wall around itself, that is called as oospore. And in the further development, that oospore gives rise to pro-embryo, embryo and pro. If you split this word, pro is the primitive stage of embryo. So primitive stage of embryo, and in the further development, then it gives rise to embryo. This is in the simplest form I have just shown you for your understanding. Okay, now we'll talk how this takes place, how this embryo is developed. Now when oospore, that means zygote, forms a wall around itself, this oospore divides mitotically in two unequal cells. Okay, now here also unequal cells are formed by the process of mitosis. So oospore gets split into two unequal cells. Larger is towards the basal side, that means what? Micropylar side. You just imagine keep the structure of anatropous ovule in front of you, so that it will be easier for you to understand. Okay, so larger cell, that is called as basal cell, which is towards the micropylar end, that gives rise to suspensor cell, whereas smaller cell is called as terminal cell, or this is present towards the chaliceal end, and that is called as embryonal cell. Now you try to understand the meaning of these cells. Okay, suspensor is having also meaning, and embryonal also is having meaning. How? First try to understand where it is located. The larger one is towards the micropylar end, okay, and the smaller one is towards the chaliceal end. Okay, so smaller one is towards chaliceal end, means this is called as embryonal cell, and the larger is towards the micropylar end, that is called as micropylar suspensor cell. Now how? We'll see this suspensor cell. Now we are talking about the cell, that is the basal larger cell, which is towards the micropylar end, that by transverse division gives rise to suspensor. Now you try to understand the meaning of suspensor, which is suspended, right? Suspended part, suspended form, that is called as suspensor. Okay, so this cell, suspensor cell, which is towards the basal, that means micropylar end, which is larger one, gives rise to suspensor. Now in the suspensor cell also, the last cell of the suspensor is large and swollen, that is called as hostorium. Whereas first cell of the suspensor is called as hypophysis. Hypo means below, physis, below that embryonal cell. So first cell of suspensor is called as hypophysis. A large cell of the suspensor is called as hostorium, which is large and swollen. Okay, I will show you in the diagram, and you will also understand it here in the uh, diagram 1.16, development of monocot and dicot embryo is given, right? So in the 1.15, that is development of dicot embryo, there you try to understand how it is taking place with the help of these diagrams. You try to understand what I am telling first, and then you look at the diagram later. I will also come to that point again, to those diagrams. Okay, so large basal cell, which is called my suspensor cell, by transverse division gives rise to suspensor, and that is towards the micropylar end. Whereas smaller cell, which is called as terminal cell, which is towards chalical end, or which is towards the, you can say, endosperm region, that is called as embryonal cell. Okay, so this suspensor pushes the embryo towards the endosperm for the nourishment. Now you will understand the meaning of this, why this is, how this is important, and how this role of suspensor cell is important. That large suspensor cell, which is working as a suspensor. What is the role of this? It is pushing the embryonal cell, that is the smaller cell which is towards the chalical end, towards what is present immediately next to the embryo. That developing embryo is endosperm, right? Just now we discussed in the middle part of the embryo sac, if you see the structure of endosperm, second fertilization result, that is endosperm is developed. And what is the role of endosperm? That is the nutritive tissue. So this suspensor cell pushes this developing embryo towards the endosperm for its nourishment. So embryo is getting nourished by the endosperm. Okay, and how it is taking place? That is with the help of suspensor. Now we'll talk about embryonal cell. Now embryonal cell, then what happens? Consecutively, 3 mitotic divisions take place in the embryonal cell, which is considered to be the terminal cell, or which is towards the chalical end, which is close to the endosperm, right? Why close to the endosperm? Because it needs nutrition, which is received from the endosperm by this embryonal cell. So 3 consecutive mitotic divisions takes place in the embryonal cell, and the result of 3 mitotic division is called as octant embryo. That is a part of pro embryo, or this is also called as pro embryo. That means first mitotic division takes place, then what happens? From single that embryonal cell, 2 cells are produced. Then second mitotic division takes place, then 4 cells are produced. Third mitotic division takes place, 8 cells are produced. You know the result of mitosis, double, it is doubled, right? So first, second, third consecutive division. So result of this embryonal cell is the number of cells which are produced are 8. Octant means 8. So octant stage of embryo is seen, and that is called as proembryo. Now out of these 8 cells of this proembryo, lower tier of 4 cells of octant is giving rise to hypocotyle and the radicle, whereas upper tier of 4 cells of the octant gives rise to plumule and 1 or 2 cotyledons. Now what does it mean? I tell you. Hypo means below. Cotyle is the word related to cotyledons. Depending on the type of species, either 2 or one cotyledons means di or monocotyledon structure. Cotyledons are present. So below the cotyledon, that is called as hypocotyle. This is and radical. Radical gives rise to roots, whereas the upper tier of four cells of this octant gives rise to plumule. That means which gives rise to in the further development shoot system. Okay, so and one or two cotyledons. Try to understand. Out of 8 cells of this octant embryo, proembryo, upper tier and lower tier, how they give rise to it is shown. Lower means lower type tier of 4 cells of this octant give rise to hypocotyle and radical, which in the further process give rise to root system. Radical gets converted into roots, and upper tier of 4 cells of octant give rise to plumule, which in the further development produces shoot system, and one or two cotyledons depending on the nature or the type of species. Either it is monocotyledonous or dicotyledonous, means if one cotyledon, monocot it is called, two cotyledons, then dicot it is called. So cotyledons, depending on the species, one or two cotyledons are produced from the upper tier of 4 cells of this octant embryo. Now what happens to this octant embryo or pro embryo? Again it develops. So in the further development, this octant embryo forms a globular type of shape. Globular structure is formed, first of all. That is called as globular embryo. Then again in the further development, what it gives? It gives heart-shaped structure of the embryo. So that is called as heart-shaped embryo. Then again in the further development, it gives rise to horseshoe-shaped structure of the embryo. And that is the last structure, where at the end, this is the development of the complete development of the embryo, where ovule is getting converted into seed and ovary into fruit. Now what we were talking everything about the ovule itself, isn't it? We were talking about the ovule, which is getting converted into seeds, and ovary, which that means ovular ovules which are present in the ovary, that ovary is getting converted into fruit. So ovule gets converted into seed and ovary gets converted into fruit. That comes under post fertilization changes. I hope you have understood all these. I will repeat in the another this thing. Now this is the diagram. See, this is the result of post fertilization. That means the zygote, which forms a wall around itself, that is called as oospore. It divides unequally. So basal cell is here in the opposite way it is shown. Means opposite direction it is shown. This is suspensor cell which is towards the micropylar end, and this is the embryonal cell which is towards the chalizial end. That means close to the endosperm. For just understanding, these diagrams are shown as they are in the orthotropous ovule, but actually we are talking about the anatropous ovule. Okay, exactly opposite you should show. This unequal base cell, that is the embryonal cell, which is smaller basal cell, that should be towards the chalizial end, and this should be the micropylar end. Okay, then this is what the further development of the these two cells. This is giving rise to suspensor, which is towards micropylar end, and this is as you know that it gives the suspensor. What is the role of suspensor cell? Suspensor cell pushes this embryonal cell towards the endosperm for its nourishment. So from the endosperm, this embryonal cell is getting nourished, and it is showing the further development. In the further development, see, this is the 4 cell structure, then octant cell structure. This is then later gets converted into globular type of embryo, and then it gives rise to the heart shaped structure and then horseshoe shaped structure. So at the end, what is formed? Seed is developed from ovule and fruit is developed from the ovary. So the goal of reproduction in every living organisms, including plants, is to create offsprings for the next generation. So seed development is initiated by fertilization. And what happens to the integuments then? That works as seed coat. So integuments get converted into seed coat. outer is called as testa and the inner integument is called as tegmen. That means outer and inner seed coats are formed. And ovule converting into seed, outer and inner integuments into seed coat, right? Outer and inner seed coat. Now there are types of seeds. What are these types of seeds? Endospermic seeds and non-endospermic seeds. Endospermic seeds are also called as albuminous seeds, where endosperm remains conspicuous and fills a greater part of the seed, called as endospermic or albuminous. Albumin is what? Protein part, right? So in case of most of the monocots as well as few dicots like castor, coconut, maize, all these plants, their seeds is of endospermic type, where endosperm is not totally utilized for the development of embryo. So it remains intact. It remains conspicuous, means in a proper proportion, or it is there present in the seed. It remains in the seed. So that type of seed is called as endospermic seed or albuminous type of seeds. Whereas non-endospermic type of seeds, they are also called as X-albuminous, without albumin. Albumin is what? A type of protein. So that's what you just remember in this way. X-albuminous means without albumin. And non-endosperm means endosperm is not there. Then where it goes, this endosperm is utilized for the development of embryo. And that's why it is not seen. Though it is present very thin and in the peppery form it is present. So that is called, this type of seed is called as non-endospermic seed, where the embryo absorbs food reserve from the endosperm completely during its developmental stages, and so endosperm disappears in mature seeds, and the resultant seed is called as non-endospermic or X-albuminous seeds. And you know, there are so many examples like pea, bean, etc. Leguminous, most of the members of leguminaceae, all these leguminous plants, they are of this type, that is non-endospermic seeds. When you soak these seeds, you can observe that papery part is separated, and that is what the endosperm part, and the insides part, that is the embryonal part, that is used for the development of embryo, and that's why it is that endosperm is not there or very in thin layer. Now, what is the significance of seed and fruit formation? Fruits provide nourishment to the developing seeds. You know that. Fruits protect the seeds in immature condition. Seeds serve as important propagating units of plants. You know, from the seeds itself, new plants are produced. So, propagating organ, that is called as. Seeds and fruits develop spatial devices for their dispersal and thus help in the distribution of species. You know that how seeds are carried from one place to another place, how the fruits are carried, and how they become the way of how they are dispersed from one place to the another place, and how the new on that particular new place again the new species can be developed like that. Okay, now the next part which is remaining from this chapter, very small parts are remaining, so that we will see structural and physiological adaptive mechanism of the survival of the seed is called as dormancy. Dormant stage, you just remember as the resting stage. Dormant form means resting form, resting stage. That means all the physiological activities, they are stopped, and each and every species, that means each and every species, they show its own dormant period is there. So for one species it may be just for few hours, for the another species maybe for several days or months or even years. So the dormant period varies from cell to cell, species to species. That period of the species is different in each one. So structural and physiological adaptive mechanism, and it is very important also, this mechanism for the survival. If the conditions are not favorable, especially environmental conditions, if are not favorable, seeds remain as it is. They will not germinate. And that's why this will this will germinate only when the environmental conditions become favorable. This is very important. Otherwise, what will happen? These seeds will be vanished from the environment. If it tries to germinate, if germinates and environmental conditions are not favorable, obviously what will it will get vanished from this surrounding and from the atmosphere, obviously from the nature itself. If it is not having this mechanism of survival, that is called as adaptive mechanism or adaptation. This is one of the types of adaptations seen in the seeds, which is very also important. So mature and viable seeds will not germinate even in the presence of favorable conditions. So their uh, dormancy period, if it is going on, then though the conditions are favorable, it will remain dormant. That means it will not germinate. Once this completion of dormant period is over, then only the seeds will germinate and then will be considered as viable. The seeds which are able to germinate only and give rise to new species only are called as viable seeds. So the dormant period varies from species to species. Now, what these few terminology, just let me discuss, then I will again come with the slides. Another slides. Now, apomixis. It is the phenomenon of formation of embryo or embryos through a sexual method of reproduction without formation of gametes and the act of fertilization. Embryo develops in the ovule and ovule develops to form seed. In apomixis, try to understand, without fertilization, there is formation of embryo. So that's why this is considered to be a sexual method of reproduction. It's not considered as a sexual method because there is no fusion of two gametes like male and female gametes. So without act of this fusion, that is the fertilization, the embryo is developed. Now, how this embryo gets developed? In apomixis, there are two ways. One is apogamy. What is that apogamy? When gametophyte organ or cell produces embryo-like structure without fertilization. That means suppose egg cell is there. Okay. That is not fertilized by the male gamete. But that haploid cell without fertilization itself produces embryo-like structure. That is called as apogamy. Gamy means gametic cell. Okay, which gives the appearance of embryo. It develops like an embryo. As it is developed from the gametophyte, that is called as apogamy. Whereas similarly, when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis, is called apospory. Apospory means when sporogenous tissue is used. Apogamy means when gametophytic cell is used, involved. Gametes are involved, or gametic cell or haploid cell is involved to produce the embryo. And when diploid cell is involved to produce the embryo like structure, which is diploid and which is not going to give rise to haploid part by meiosis. Okay, so gametophyte is developed, but that is not haploid but diploid. So that is called as apospory. Always remember, gametes are always diploid and somato or sporogenous tissue is always diploid. Gametes are always haploid. Gametophytic cells are haploid in nature, right? So this is what spatial feature, and that is observed in orange, mango, etc. So apomixis, it is of two types. Apomixis is nothing but the formation of embryo without fertilization, right? When it is formed, the embryo-like structure is formed from the gamete, then it is called as apogamy. When it is developed from the sporophytic cell, then it is called as apospory. Only these points you need to remember. Now again, there are categories of apomixis. You try to understand, though it's not in detail, but I'll just mention it. Recurrent apomixis means the embryo sac generally rise either from an archisporial cell or from some other part of the nucellus. Now, if you remember the structure of anatropous ovule or the ovule, angiospermic ovule, you can say, then at the center, what is present? Embryo sac, which is surrounded by nucellus, that is the nutritive tissue. If the embryo sac generally rise either from an archisporial cell or from some other part of the nucellus, then that is called as recurrent apomixis. Means what? Embryo sac is developed either from the archisporial cell or from some part of the nucellus. Means how this embryo sac is developed from archisporial cell, or it is you don't, I will not go in detail about this part, but just remember nucellus, you know. So from the one of the cells or some part of the nucellus, when it gets developed, that is called as recurrent apomixis. Now what is non-recurrent apomixis? Embryo arises either from the egg by parthenogenesis or from some other haploid cells of gametophyte through apogamy. And plants produced by this method are generally sterile and do not reproduce sexually. Nicotiana is the example. That means what? Embryo is produced from the egg, just like apogamy I was telling in the first type. In the different ways I was telling about the apomixis. That means from the egg cell. Parthenogenesis means without fertilization. Embryo is produced from egg cell, right? So that is, or from even other haploid cells of the gametophyte. Whatever the gametophyte is produced, any cell of the gametophyte or egg cell, then that without fertilization when embryo is produced, or then that type of plants which are produced by this method are generally sterile because there is no involvement of the another gamete over here. So they do not reproduce sexually. So that these are considered to be sterile. And the third category of this is next way. Now, next terminology we will talk about, that is the adventive embryony. What is that adventive embryony? Embryos may develop from somatic nucellus or integuments along with normal zygotic embryo. Now try to understand what exactly it means. Now in this adventive type of embryony, normal type of embryo is formed. What is the normal type of embryo? The result of fertilization. That means male and female gamete egg cell is when fuses with the male gamete, the result is zygote. So from the zygote, in the normal way, embryo is produced. That is called a zygotic embryo. Along with that, when the embryo may develop from somatic nucellus also. Nucellus, that means the embryo sac which is covered by nucellar cells, or even that nucellus is covered by integuments, you know, outermost layers, one or two integuments. So either from nucellar cell, which is somatic cell, means diploid in nature, as well as new integuments also diploid in nature. When the embryo is developed from somatic nucellus or integumental cells along with the normal zygotic embryo, then that type of embryony is called as adventive type of embryony. And the examples are mango, orange, lemon, etc., which gives rise to a condition called polyembryony. That means the normal embryo is developed, and along with that, from the nucellar cells or integumental cells, few more embryos are also developed by parthenogeny. That means without diffusion, without fertilization, right? That is called as adventive type of embryony. Now what are these types? Parthenocarpy. The fruit is developed without the process of fertilization, that is called as parthenocarpy. The development of fruit without fertilization is called as parthenocarpy. Examples: pineapple, banana, papaya, etc. All the seedless fruits, these are examples of parthenocarpy. Without fertilization, these fruits are developed. And polyembryony is the condition, that is the development of more than one embryo inside the seed. And the example is citrus. So polyembryony increases the chances of survival of the new plants. So though one embryo is destroyed, not developed, there are some other embryos also for its development, so survival chances increase. So this is all about the terminologies used here. I will not talk about the questions right now, but I will show you the same part with the help of a few slides, which you can see. Post fertilization changes here. What are these structures? You can see in the diagram. See here how it is getting converted into, this is the innermost part, that is the ovule. That ovule is getting converted into seeds. You can see here, these are the seeds. So many ovules, so many seeds. and this part, the ovary, integuments, that is the seed coat. So ovary getting converted into fruit, right? So this is the the post fertilization events you can see here in these diagrams. And this is absent in such families like Orchidaceae. Okay, and you can see that some other examples also. Diploid endosperm is found in Oenothera lamarcina. Then stony endosperm is present in betel nut, areca nut. You know that the pictures are given here, and also date palm. Then mature seeds are of two types, as I told you: non-endospermic or ex-albuminous seeds. Here are some of the examples where endosperm is completely consumed by the developing embryo. So these are some of the examples. You know, these chane, herbera, green peas, watane, these are all leguminous plants, most of these. Groundnut, these are examples, right? Then endospermic seeds, where cotyledon endosperm is still there. Embryo does not consume all endosperm, but it persists over there in mature seeds. And these are called as endospermic or albuminous seeds. And that food is stored in endosperm only. So wheat, maize, barley, sunflower, coconut, these are some of the examples. Okay. Then embryo development, that is called as embryogeny. Embryo is generated, formed. So in case of dicot, you can see two cotyledons, and the similar type of structure is seen in case of monocot, the same way, but only instead of two, only single cotyledon is developed. So these are the stages, early stages of embryogeny, which are similar in both dicot as well as monocot. See, these are all the structures as I showed you. Egg cell, after the fusion, that it's zygote. Zygote forms a wall around itself, that is oospore. Then again unequal two cells produced, and that is the larger one and the smaller one. Larger is towards the basal side. Now here is the correct sequence and the diagrams shown in these pictures, in these all diagrams. Suspensor is towards micropylar. See, longitudinal transverse division of suspensors, just like a plate, isn't it? Hair plates, it looks like. And this last cell of this suspensor is called as hostorium. And the this one is which is close to the embryonal cell. This is the first cell of embryonal cell. So hypophysis, that is called as. So this is the two cell structure. This is the octant stage. That means, though you can see four here, four are to that side. So this is the octant form, and this is the entire structure of suspensor. Okay, this is called a suspensor. So this, what is the role of suspensor? It is pushing the embryonal cell towards the endosperm. Okay, now this octant stage gets converted into globular. This is the further development, then heart-shaped structure, and then horseshoe-shaped structure. So in this octant stage, upper tier and lower tier of cells, how we discussed that, you remember, lower tier gives rise to hypocotyls and radicle, which further development gives rise to root system, whereas upper tier of cells gives rise to one or two cotyledons, and then plumule. Plumule gives rise to root system. And in the further development, at the end, when the role of suspensor is over, it gets what you can say, diminished from there. So its role is over, then it gets degenerated, suspensor cell, in the later stage. So this is what the two cotyledons here, in case of dicot, two cotyledons, and in case of monocot, single cotyledon. Rest of the things are similar. The same thing I told you, that see, this is the you can see now, these are the two cotyledons. This is the plumule, which is the upper part, and which gives rise to shoot system, and this is the radicle, which is covered by the root cap, that gives rise to root system, and this is the hypocotyle. Hypo means below the cotyledon, so this is called as hypocotyle. Okay. Then monocot embryo, little just 1.212 points you just remember. That is most of the members of grass family, or graminy also you can say. Cotyledon, it shows the scutellum, that is situated towards one side, lateral side of the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cap is enclosed in an undifferentiated shape called as coleorhiza. Coleorhiza and coleoptile, these are the two terminologies. Coleorhiza is that covers radicle, that in the further process gives rise to radicle, and coleoptile, that is giving rise to plumule. So the portion of embryonal axis above the level of attachment of the scutellum is called as epicotyle. Epi is above, hypo is below. You remember always. Just these 2 points are different. Now see, this is the structure of monocot embryo. Here is the scutellum. This is the coleoptile and this is the coleorhiza. Coleorhiza, which gets converted into root system. That means root cap, that radicle, is covered by coleorhiza, whereas plumule is covered by coleoptile. Okay, so coleoptile, in the further development, it gives rise to shoot system, that is the shoot apex here, and this is the root cap and the radicle covered by coleorhiza. In the primitive stage, that these are very delicate, so these are covered by coleorhiza and coleoptile. Now, next part is not there now for your this is what you that is the end of this chapter. We have covered almost everything. I will show you few pictures of the seeds also. Now see seed, how it looks. uh, Fertilized ovule. See, when you take the longer section, a longitudinal section, how it is seen. This is the embryo, this is the cotyledon, and this is the seed coat, isn't it? This embryo is getting converted into the new plant. See, this is what. And the outermost covering of the seed is seed coat. It has 2 layers. Outer is called as testa, inner is called as tegmen, and that hilum is a scar on the seed coat through which the developing seed were attached to the fruit. That is the point hilum. You know, you have studied the structure of anatopous ovule. Above the hilum is a small pore that is called as micropyle, and within the seed coat, the embryo consisting of an embryonal axis and 2 cotyledons, and that cotyledons are fleshy and full of reserve food material, which are used for the development of embryo. See, these are the developmental stages of the seed. How this development after the germination, what is happening. Seed coat it is. This is the plumule giving rise to shoot system, and this is the radical axis, which in the further development gives rise to root system. This is the seed coat with the 2 cotyledons. See, these are the cotyledons. Radical giving rise to root system, and plumule which gives rise to shoot system. Okay, so this is all about the types of seeds, and this is the structure of monocot, dicot. Again, this is shown what is pericarp, endosperm, scutellum part, which I already discussed. I'm just showing you these pictures. You know, seed dispersal means what? Seeds can be carried away, dispersed by many agencies like water. Now you can see that coconut, how it is developing. It is by agency of water from in the sea. You can say from one place to another place, and there then the new on the new area that species is developed, isn't it? So these are this is what seed dispersal. There are different types of seeds, and accordingly by different agencies these seeds get dispersed from one place to another place. See, these are few pictures. The birds, water, insect, wind, these are some of the ways, agencies. So this is what about the seed dispersal and all. So this is the end of the chapter. Now I'll stop my presentation.